Chapter 16, question 10, gives you the primary structure of a peptide. That's the order of amino acids, so leucine, valine, cysteine, aspartic acid. And then it asks you questions about the tertiary structure. So, first thing we want to do is to classify each of these amino acids. And we can do that by referring to the table. So here, leucine, Leu, is our first amino acid. And if you look at the table, you'll see that leucine is right here, and it's a non-polar amino acid. So we can classify that as non-polar. Okay, valine. If we go to the amino acid table, table 16.2, we see that valine is over here, and it also is non-polar. So we can say valine is non-polar. Now cysteine is a really special amino acid. We saw in chapter 16, question 9, how cysteine forms those really strong disulfide bonds. Sulfide bonds. That's what we want to watch out for cysteine. And if you look at the structure for cysteine, it is a polar amino acid, but it has that sulfur on the end. So another cysteine can come by, face the opposite way, right? You have the backbone there. And these two SHs can snap together to form a bond between the sulfurs. And that's the disulfide bond. So that's just a special thing that only cysteine can do. So that's what we want to watch out for with that. And then ASP, that stands for aspartic acid. Aspartic acid is down in the bottom left, over here. And you can see that that is a polar amino acid. Remember, polar is everything from the middle down. And not only that, but it's acidic. Okay, so I'll put polar there. So these are the characteristics of these individual amino acids that's going to affect how the chain folds globally. In other words, this is the characteristics of the primary structure that are going to affect the tertiary structure. So part A asks you which amino acid can form a disulfide bond. Well, that is clearly cysteine. So you'd have cysteine here. That's what we want to know. When you see cysteine, immediately you'd associate that with the ability to form those really strong disulfide bonds. So you'd say cysteine. Next, which amino acids are likely to be found on the inside of the protein structure and why? Remember that these proteins are going to be in your body fluid, it means they're going to be surrounded by water. And so what they'll do is they will take the backbone and fold so that all of the non-polar amino acids sort of face the center so that they're with each other, and the polar amino acids face the outside. So, like dissolves like, non-polar R groups will want to face each other rather than the polar water. So, for these, you'd say, which of these are non-polar? Well, we saw that leucine and valine are non-polar. So those are going to be those amino acids in the center. So which ones are likely to be found on the inside? Leucine and valine are likely to be found on the inside of the protein because they are non-polar and the outside of the protein is facing polar water. So that's why they would not want to face the outside of the protein. Instead, they would face the inside. For C, it says which amino acids would be found on the outside of the protein. Well, we just saw how the outside of the protein is surrounded by polar water. So what's going to be surrounded by polar water? Polar amino acids. So we saw here that aspartate is polar. And so it would want to be on the outside. And also, if you noticed, we pointed out that even cysteine is polar. It's in this polar section. So those two polar amino acids would be on the outside. So aspartic acid and cysteine would be found on the outside of the protein because they're both polar and water is polar, and like dissolves like. D says, how does the primary structure of a protein affect its tertiary structure? Remember that primary structure is the order of amino acids in a chain or in a peptide. Remember the difference between a peptide and a protein. A peptide is usually less than 50 amino acids, and then a protein is usually 50 or more, but that's just a rule of thumb. So here they say, how does the primary structure affect the tertiary structure? The order of amino acids is the primary structure. Here we have valine first, or leucine first, then valine, then cysteine, then aspartic acid. If we were to put aspartic acid first, that would change the order, and so it would change the primary structure. The tertiary structure is how that chain of amino acids, so how a peptide folds globally. So the whole big thing, not just wrinkling in local places, but the whole big thing folding over. And what we saw in this problem is that the primary structure, we saw that, we saw that where amino acids are placed in a protein, that's its primary structure, right? The order that they're in. Determines what part of the protein will stick together through hydrophilic interactions, hydrophobic interactions, and disulfide bonds. That is its tertiary structure. So where amino acids are determines what parts of the chain come together. And that's how the primary structure of a protein affects its tertiary structure.